Good day, my dear students. Today we are in for another class, and in this lecture series, we are going to be talking about protein. And under protein, we'll have the definition of protein, protein structures, classification of protein, amino acid catabolism, ammonia transport in the blood, urea cycle, and transamination. These are the topics we are going to be discussing under protein. I wish you a happy listening and be part of the discussion. Thank you. Well, like I said, we are going to be talking about proteins. And first, essentially, we have to start with the definition of protein, or what protein is all about. Protein is a micronutrient that is essential to building muscle mass. It is commonly found in animal products, though it is also present in other sources such as nuts and legumes. There are three macronutrients, and one of these macronutrients is the protein. Macronutrients provide calories or energy. Then after that, we're going to look at protein structures. And in proteins, we are going to look at protein structures, the types of protein structures we have. We have the primary structure, which is a succession of amino acid residues, usually abbreviated by the 1- or 3- letter codes. Then you have the secondary structure, is the 3D arrangement of the right-handed alpha helix, or alternative structures such as a beta pleated sheet. Then you have the tertiary structure, is the 3D folding of alpha helix. And they also have the quaternary structure, is the arrangement of these subunits. So in hemoglobin, as shown, the quaternary structure comprises of alpha and 2 beta polypeptides held together by electrostatic. So basically, these are what we're going to be discussing under protein structure. And I have for you here examples of such structures, and finally the summary of what these structures are all about. Then we go to protein classification of proteins, and we'll look at how proteins, what are those things that were considered under the classification of proteins before proteins are classified. We have a summary here. Proteins can conveniently be grouped on the basis of both physical, that's the shape, and chemical properties. The physical properties commonly employed for grouping are those of solubility and heat coagulation. Standing on these two characteristics, proteins are classified into three as simple conjugated and derived proteins, which you can see here. So this classification is by solubility and composition. I have a very broad summary and the involvement of the various amino acids. Then simple proteins, which comprises of all the examples on the chart. We'll briefly discuss them one or the other in a class proper. And also you look at the conjugated proteins, mucoproteins, chromoproteins, hemoglobin, cytokine, all under conjugated proteins. Then you have derived proteins, where we have primary proteins, we have metaproteins, coagulants, and secondary proteins, you have proteases or albumosis, tones. So that is that about the classification of protein. We're going to talk about it in details when we meet in our class proper. Then our next topic will take us to amino acid catabolism. We'll be discussing about amino acids, and as you all know, they are produced by the amino acids are produced by catabolism. So they may be directly recycled to form new proteins, converted into different amino acids, or can undergo amino acid catabolism to be converted to other compounds via the Krebs cycle. So oxidative deamination is the first step to break down the amino acids so that they can be converted to sugars. The process begins by removing the amino group of the amino acid. So the amino group becomes ammonium as it is lost and later undergoes the urea cycle to become in the liver. So here is the flow chart of the amino acid catabolism. We're gonna discuss more of this in our class proper. So next we'll be talking about ammonia transport in the blood. After it is being produced, how is this ammonia transport in the blood? Ammonia is loaded via glutamine synthesis by reaction. NH3 plus glutamate will give glutamine. It occurs in nearly all tissues of the body. Ammonia is unloaded via glutaminase by reaction. That is, glutamine will give ammonia plus glutamate. It specifically occurs in kidneys and intestine, and in very low concentration. Here you see the pattern, the chain of, and we'll discuss more seriously about it in our next class, and that will lead us to our next topic, urea cycle. Like I told you, the urea cycle is a mechanism for removing unwanted nitrogen. Urea formation in the liver starts with the multi-step conversion of ornithine and arginine to arginine. This is followed by the breakdown of arginine into ornithine and urea. The complete urea cycle, as it occurs in the mammalian liver, requires 5 enzymes. We have arginine succinate synthase, arginase and arginosuccinate layers, which function in the cytosol, and the ornithine transcarbamylase and carbamyl phosphate synthase, which function in mitochondria. Additional specific transport proteins are required for the mitochondrial uptake of L-ornithine, ammonia and hydrogen carbonic acid, and for the release of citrulline. So the stepwise reaction that we're going to be discussing in our next class, and that will lead us to our next topic, transamination. We will also look at transamination. What is transamination? Is the process by which amino groups are removed from amino acids and transferred to acceptor, keto acid, to generate the amino acid version of the keto acid and the keto acid version of the original amino acid. Transamination is amino transfer reactions. So transamination reaction combine reversibly amination and deamination, and they mediate redistribution of amino groups and acid. So we will be looking at that, and this is the reaction of how it takes place. Transaminases, aminotransferases, are widely distributed in human tissues and are particularly active in heart muscle, liver, skeletal muscle, and kidney. The general reaction of transamination given there. So transamination, the alpha ketoglutarate / L-glutamate couple serves as an amino group acceptor / donor pair in transaminase reaction. So this is it for the complete reaction. And with this, we will be in our next class. So I will want you to go through this and use it to guide yourself pending when we meet in our next. Thank you for listening.